We all know that our bodies have an amazing ability to heal themselves. In particular, skeletal muscle has a large regenerative capacity. Today I want to tell you about my research, which investigates how this regenerative process occurs in skeletal muscle and why understanding this process more thoroughly can lead to the introduction of new therapies that can enhance muscle regeneration. Previously, scientists have identified different cell types and their roles in muscle regeneration following volumetric muscle loss. Using powerful imaging techniques, the roles of muscle stem cells and fibroadipogenic progenitor cells, aka FAPs, have been uncovered. Following injury, FAPs secrete trophic factors, which stimulate muscle stem cells to regenerate the muscle fiber. These FAPs are then cleared through programmed cell death. However, the case with chronically damaged muscle tissue is that the FAPs survive by evading cell death. The result is that FAPs then differentiate into fibroblasts and fat cells, forming scar tissue, which impairs the muscle's ability to regenerate. This process is known as fibrosis. We can see this play out in the image at the top left. Looking at a mouse's normal TA muscle, we see regular muscle histology and an intact laminin, as indicated by the green staining. However, the tile below shows what happens following volumetric muscle loss. Our laminin is no longer intact, and looking at the yellow box, we see fibrosis occurred following attempted muscle regeneration. The purpose of my research is to better understand the spatial cellular dynamics in skeletal muscle following volumetric muscle loss. In order to do this, I am using multiplexed imaging technology called co-detection by indexing, aka codex. We can see an example of numerous codex images on the right. Codex allows us to visualize up to 60 proteins on single sections of skeletal muscle using antibodies labeled with unique DNA barcodes that are imaged 3 at a time in an iterative process. By examining numerous secretive factors, we can identify the cells upon which these factors act, ultimately allowing us to get a better understanding of the microenvironment of skeletal muscle and which cells and proteins play a role in the regenerative process. This research is important because by understanding muscle regeneration and what triggers fibrosis, we can find ways to enhance skeletal muscle's natural ability to regenerate. For example, using scaffolds in conjunction with muscle stem cells can lead to muscle stem cells differentiating into muscle fibers, thus repairing the damaged tissue. In conclusion, by using codex to understand the cells and the key proteins involved in muscle regeneration, we can gain insight into what triggers fibrosis and ultimately prevent it, thus opening the door to revolutionary therapies to repair muscle. Thank you.